Hello, my name is Fiaz Shah, and I'm a senior in the Math Science Technology Center at Paul Lawrence Dunbar High School. And today I'll be presenting on my capstone research project completed under the mentorship of Dr. Brian Delisle at the University of Kentucky Department of Physiology. My project is titled Improving the Clinical Diagnosis of Long QT Syndrome. So to start, I'll be giving a brief overview of what I'll be discussing throughout this presentation. So I'll be starting with an introduction, then I'll move on to the problem and goal, the question and hypothesis, variables, methodology, results, conclusion, future directions, acknowledgements, my references, and then I'll leave my contact information if there's any questions that you might have. So to give a brief introduction of what long QT syndrome is and why it's important. So long QT syndrome is an inherited disease that could cause deadly arrhythmias. Now, an arrhythmia is just a general term for any sort of disruption in the normal heart rhythm, such as a skipped beat or an extra beat, and that can greatly increase a person's risk for sudden cardiac death. Now, of course, cardiovascular diseases are among the leading causes of death in the world, and it's especially important to me because I have a family history of sudden cardiac death, and I wanted to help patients avoid this risk because it's so unpredictable, it's sudden, and that makes it really scary. So to put long QT syndrome into perspective, this diagram right here on the right side is showing an electrocardiogram or an ECG, which displays the electrical activity of the heart. So each one of these waves is sort of representing a specific event, so to speak, in the muscle contractions in the heart that are producing heartbeats. So this right here is the P wave, then the Q wave, and then the peak is the R wave, and the S wave, and the T wave. So the distance between the Q wave and the T wave is going to be the QT interval. And as you can see, a person who has long QT syndrome right here has a little bit of a longer QT interval. Now, what that means is there was a delay in the repolarization of the bottom two chambers of the heart. So that's shown in this left diagram right here. This is the right ventricle and the left ventricle. And if there's a delay in essentially the relaxation of those muscles, then there's going to be an inefficiency in the pumping of blood that could lead to things like sudden cardiac death. And I also do want to point out the peak or the R wave. The distance between two R waves is going to be the RR interval, and that's essentially the same thing as heart rate. So I'll be using RR interval and heart rate interchangeably throughout this presentation. Now, the QT interval is dependent on heart rate. It's going to naturally get a little bit longer as heart rate decreases because there's going to be a bigger gap in between beats. So how are clinicians supposed to be able to identify if a patient's QT interval is actually prolonged? Because it could just be relative to a slow heart rate. And so that's where Bazett's QT correction or QTC formula comes in. It's the QTC equals the QT interval over the square root of the RR interval, where the QT interval and the RR interval are measured in milliseconds. So what this formula does is it essentially calculates a standardized QT interval to see what it would be at a standard heart rate, and it'll correct for that dependency on heart rate so the clinician can actually assess if a patient has a longer QT interval than it should be. And because of that, this formula carries heavy weight in diagnosis. Now to move on to diagnosis, to describe it in one word, it's really tricky and it's really subjective. It's currently carried out using a Schwartz scoring system where basically a certain number of points is given to a patient depending on factors like that calculated QTC or other phenotypic symptoms. And by the end of the test, the clinician adds up the points and basically sees how many points they have and corresponds that to a probability of having long QT syndrome. So clearly it's up to misinterpretation, and that's why a previous study found that 2 in every 5 patients that were diagnosed with long QT syndrome did not actually have the disease. So that's a huge problem. That's about 40% of people who were misdiagnosed. There could have been groups of people that were overdiagnosed, and that created a huge issue. And so clinicians decided to start looking solely at genetics because long QT syndrome is inherited. And they decided, okay, if you have a mutation that causes long QT syndrome, then you must have the disease and we're going to automatically get you started on treatment. And the problem with that is that just because a person expresses a mutation doesn't mean they're going to have the disease. And what the clinicians started doing to treat these patients was putting in ICDs, which are implantable cardioverter defibrillators, into patients that didn't necessarily have long QT syndrome. And what's even worse is that they were putting these into kids who, had their whole life ahead of them, and they're going to suffer the side effects of this treatment. And not only is it just a misinterpretation at this point, it's an invasive procedure that's almost doing the opposite of helping these patients. So diagnosis of long QT syndrome is really tricky, which leads me to the problem. It all goes back to Bazett's formula because it carries such heavy weight in diagnosis. And essentially, if I go back to the formula really quick, this square root is there for a reason, this fraction is there for a reason. It's showing the presumed relation between the QT and RR interval. But what I'm proposing is that it doesn't display the true relation between the QT and RR interval. And instead, it relies on a one-size-fits-all sort of principle, which is what's causing all these misinterpretations and diagnoses. And instead, the formula should be tailored to fit a specific individual's QT and RR relation so that a better corrected value can be outputted and considered in diagnosis. So the goal, of course, of this project would be to improve the diagnosis of long QT syndrome by proposing a more precise way to correct the QT interval. And so for my formal experimental question, I'm looking at I looked at how does the QT interval change over a wide range of heart rates or RR intervals in mouse models. I wasn't able to test this on humans, of course. So I hypothesized that the QT and RR relation in mice with long QT syndrome is fundamentally different than that of normal healthy mice. So I had 12 total mice, which I'll discuss in the variables. So my independent variable is what I'm changing in this experiment, and that's going to be the type of mouse model. So I had a group of 6 long QT syndrome bearing mice versus a group of 6 control mice. So 12 total mice. And of course, my dependent variable, what I'm measuring, is going to be that QT and RR relation via a linear regression or a slope analysis, which I'll discuss a little bit later. So for my methodology and how I went about carrying out this experiment, it can be divided into 3 distinct parts. To start, first I had to be able to monitor the electrocardiogram of each mouse, so I had to learn how to implant a telemetry system into the mice. Then I needed to analyze the data on a software called Panima. And then arguably the most important part was to transform Bazett's formula. So I'll go in depth in those 3 little parts. And so the ECG telemetry implantation started out with the mice being anesthetized with isoflurane, and then incisions were made near the abdomen of each mouse. And basically this little transmitter unit, which I'll show in a picture in a second, and it was implanted into the peritoneal cavity, which is right below the stomach, and 2 ECG leads, one positive and one negative one, were attached to the respective areas, and then that transmitter was able to wirelessly send that data to a receiver that I can hook up to a computer to analyze the data. So this diagram right here, the one on the left, is showing what the transmitter unit looked like. It had a positive and negative lead, and it was basically routed to, the negative lead was routed to the right acromion, which is right below the shoulder blade, and then the positive lead was routed to the apex of the heart. So this cycle of charge basically allows the transmitter to detect the electrical activity of the heart, then wirelessly send that information so that I can analyze it. So the next part would be to analyze this data on Panima. And so what Panima does, it shows me electrocardiogram, just like I showed you earlier, and it basically places marks at the specified ECG wave. So I was interested in looking at the Q wave, the T wave, and the R wave. So it places marks to the best of its ability at those areas, and then it generates a list of numerical parameters. So those numerical parameters are able to calculate the QT and R intervals in milliseconds, of course, based on where those marks are placed. And so it was a very tedious process because I had to go through hours of ECG footage to find sort of good marks, because the computer is not accurate most of the time. It places really bad marks. So I discard those marks in analysis. I make sure to pick good marks, and I did about 150 pairs of QT and R intervals across 5 ranges of heart rate for each mouse. So I did want to account for all sorts of heart rates that the QT interval could change at. it was 200 beats per minute to 700 beats per minute because mice's heart beat a lot faster than humans. And then I copied those marks to Excel so that I could later plot them. So this is essentially what I see on Ponema. It shows an electrocardiogram, and then it places the marks at the respective waves, and then it can calculate them, and then I can use those as quantified data. So the last part is a transformation of Bazett's formula, and essentially this, what I showed earlier was the QTC equals the QT interval over the square root of the RR interval, Bazett's original formula. And what I've done is I've made the 1/2 power, the square root, into a variable x. Now, using natural logarithm and properties of natural logs, I was able to transform this formula into a linear function. And so this variable x essentially becomes the slope when I plot the RR interval versus the QT interval, and that is what shows me the precise relation between the QT and RR interval for that specific mouse in this case. So this formula is going to be really important in how the QT interval is going to be corrected more accurately to fit a specific patient. So for my results, I first wanted to look at combining the data. So I combined all the data of QT and RR intervals across all the 12 mice in both groups. So this graph right here, the RR interval is going to be in the x-axis, and then the y-axis is going to be the QT interval. I'm seeing how that changes as a function of heart rate. And so the red line right here is the long QT syndrome group, and the gray line is the control group. So you can see there was a substantial difference in the slopes. The long QT syndrome group had a much steeper slope of about 0.8 versus the control group with a slope of about 0.2772. Now, I also did want to look at some individual data. So I looked at the average slopes for each of the mice, and I found that the average, shown here on the left, of the long QT syndrome group was about 0.829 versus the control group being about a 0.273 average. And each one of these points is representing where each of the 6 mice fell according to the slope. So all 6 mice had steeper slopes than all 6 of the control mice. I did also conduct an unpaired t-test of the difference of means, and that yielded a p-value less than 0.001, which means the data was extremely statistically significant. And then this graph on the right, I also did want to look at the R-squared values to sort of see the strength of the data. And so the long QT syndrome group had a little bit of stronger data than the control group, with a R-squared value of 0.7716 versus the control group being 0.4788. And so that leads me to conclusions and a discussion about what these results mean and why they're important. So the steeper slopes in the long QT syndrome group means that the QT interval changes more greatly as a function of heart rate. As heart rate increases, the QT interval increases by a pretty substantial amount compared to the control group. And the greater R-squared value basically means that more of the variability in the QT interval that was shown in the data was explained by heart rate, which is a good thing, and it means that the data was strong in the long QT syndrome group compared to the control group. Now, more conclusions that can be drawn from this data is that it did support my hypothesis. I hypothesized that the QT and RR relation was fundamentally different between the two groups, and that is exactly what I saw because one of those slopes were much steeper. And that's why a similar analysis should be done on patients to output a more fitting QTC value that's tailored to fit a specific patient to be used in diagnosis so that the patients can be treated as early as possible. Now, recently, this has actually become recommended by the American Heart Association, actually after I started this project. And clinicians don't know why they should be implementing this sort of analysis. And I'm hoping that not only will this project show a better way to correct the QT interval, but also to show clinicians why they should be implementing this same analysis, because that QT and RR relation is so different in patients with long QT syndrome. So in terms of future directions, I'd really like to conduct some heart rate variability analysis to shed some light on the arrhythmias associated with long QT syndrome other than just a prolonged QT interval. Now, heart rate variability actually is a direct indicator of sympathetic nervous system activity. So that'll involve the brain to this project a little bit and show me about more arrhythmias. And to actually implement this project, humans, I think the best way to do that would be with a system of Holter monitors. So a Holter monitor is essentially a portable ECG device that can be attached to a person. And if a clinician basically attaches a Holter monitor to a patient, then they can go about their day while this little device tracks their electrical activity of the heart. So the reason why it's so good is because it'll track the electrical activity as it is on a normal basis, whether the person goes to work, they go to school, whatever their diet is, if they exercise or whatever. And then they can report back to their clinician and then assess that QT and RR relation and make a better and more accurate diagnosis to treat the patient if needed. So I'd like to make some acknowledgements. This project would not be able to have been done without the help of my mentor, Dr. Brian Delisle, who I really appreciate for allowing me to work in his lab and having to put up with a high school student, even though he's a very, very busy guy. And I also would like to appreciate, I also appreciate the members of the Delisle lab, such as Dr. Elizabeth Schroeder, who helped me with my science fair presentations, and also Tanya Seward for helping me learn the mouse implantation procedure. I also like to thank the University of Kentucky and KYRP, the Kentucky Young Researchers Program, for making this all possible, and as well as Miss Young for being really, really helpful in this whole process and making research something that's really going to help us in the future, as well as, of course, the MSTC program for placing such a huge emphasis on these sorts of things. And I'll also like to thank my friends and family for supporting me throughout this journey. Here are some of the references that I used throughout my project and this presentation. And if you have any questions about anything that I talked about long QT syndrome, about the diagnosis, or implementing the procedure, or anything, you can contact me. Please feel free. My email is sfshah2001@gmail.com. I did number the slides, so you can ask me about a specific slide or anything that you have questions about. So thank you so much for listening, and I hope you enjoyed my presentation on my research project.